Hi, I am Cheyenne Lupton, and this is my first video for Exam 4. This is on point mutations in nucleic acid sequences as well as their effect on organisms. A coding strand of DNA is shown below. The translated version of the normal mRNA is this translates to the amino acid sequence, and this will be the normal sequence. A point mutation is a change in a single nucleic acid in the sequence. There are two types of point mutations. A transition changes a purine to another purine or a pyrimidine to another pyrimidine. Examples of this are an A to a G or a C to a T. A transversion is when a purine is changed to a pyrimidine or vice versa. Examples of this are a C to a G or an A to a T. There are many effects a point mutation can have. The first type of point mutation is a missense. This is when the mutation changes the amino acid that is coded for. For example, if the DNA sequence is changed to this, or an A is changed to a T, this would be considered a transversion, and the translated sequence would be this. Because of this, the amino acids would be, as you can see, the third amino acid has been completely changed. Now the sequence codes for something completely different, which could be very bad for the organism. Many times when this happens, the protein functions less effectively. The next type of mutation is a nonsense mutation. This mutation changes the amino acid to a stop signal. For example, using the original DNA coding strand, if the sequence was changed, where the G was changed to an A, this would be a transition. Be this, which results in these amino acids. Only the start and mutated protein were coded for. None of the following proteins were coded for, meaning the organism won't get the other amino acids needed to function. Another type of mutation is a silent mutation. This mutation is least problematic. A nucleic acid is changed, but the codon ends up coding for the exact same amino acid. For example, if the DNA coding strand was this, where the T is changed to a C, which is a transition, the translated mRNA would be, and the sequence of amino acids would be this. The amino acid sequence is the exact same as the original because GCC and GCU both code for ALA. This point mutation causes the least amount of problems, and usually it is not known when this happens, which is why it's called a silent mutation. The last type of mutation is a read-through. A read-through changes a stop codon into an amino acid. An example of this is shown below, where the T is changed to an A, which is a transversion. As a result, the mRNA and amino acid sequences would be this. The stop is turned into an amino acid, so the following amino acids would also be coded for. This could be bad because more amino acids would be coded for than needed. Overall, all types of point mutations can be bad, and it is important that we have regulation in genes to keep this from happening all the time.